Imagine a 100-year-old woman. She's sitting at her kitchen table on a Sunday morning, and her memory is just razor sharp. She's cognitively entirely intact, effortlessly finishing the New York Times crossword puzzle. But if you were to take a scan of her brain, it reveals this absolutely terrifying picture. I mean, it is completely choked with Alzheimer's disease.
Right, yeah. Structurally speaking, her brain is loaded with these toxic plaques.
Yeah.
By every established rule of modern medicine, she should have severe, debilitating dementia.
But she doesn't. And, for you listening, that is the ultimate question here. How is that even possible?
It's fascinating because it forces us to completely rethink what it means to, actually have a disease. Usually when we talk about a medical diagnosis, there's this comforting precision to it.
Right, like a clear cause and effect.
Exactly. You fall down, you get an x-ray, you see a jagged white line on the bone, and the doctor says, well, your arm is broken. The structural damage directly equals the functional problem.
And that expectation just completely shatters when we step into the world of neurodevelopment and aging. I mean, this 100-year-old woman's brain is structurally full of disease, but functionally, perfectly healthy. It's basically the ultimate medical paradox.
Which is exactly why today's source material is so vital for us to dig into. We're doing a deep dive into a really groundbreaking paper from Nature Medicine, published in June 2026.
Yeah, and the researchers set out to solve this exact mystery, right? By comparing the brains of two very distinct groups of people.
They did.
Yeah.
First, they looked at octogenarians, so people in their 80s.
Yeah.
And within that group, some individuals were living with dementia, while others were cognitively healthy, despite having brain pathology.
Right, and then they compared those 80-year-olds to a group of exceptional centenarians, people over 100 years old who aged with this profound cognitive resilience, even though their brains were, frankly, riddled with Alzheimer's pathology.
And the overarching mission of this study in our deep dive today is to map the exact microscopic inflection point where the brain tips.
The inflection point, I love that term.
Yeah, we want to find the precise moment a brain goes from successfully adapting to Alzheimer's pathology to ultimately succumbing to it. Because understanding that inflection point shifts our focus from just looking at what goes wrong in the brain to fundamentally understanding what goes right in these incredibly resilient centenarians.
Okay, let's unpack this, because to really appreciate why this new research rewrites the rule book, we have to establish the old rule book first. So for decades, how has the medical field traditionally understood the progression of Alzheimer's?
Well, the field has relied on what we call the classical ATN staging model. Think of it as this linear, inevitable cascade. First, you have A, which stands for amyloid-β.
Right, the sticky stuff.
Exactly. These are sticky protein fragments that, instead of being cleared away by the brain's garbage disposal systems, they misfold. They clump together outside the brain cells, forming these dense, toxic plaques. So amyloid is step one.
Let me guess. A is for accumulation of amyloid. So what triggers step two, the T?
That's a good way to remember it. So according to the classic model, those amyloid plaques eventually trigger T, which stands for tau, specifically toxic tau tangles.
Now, tau isn't inherently bad, right? Like, we all have it.
Oh, absolutely not. Normally, tau is a super helpful protein inside your neurons. It acts like railroad ties, stabilizing the microtubule tracts that transport nutrients up and down the cell.
But in Alzheimer's, something goes wrong with the tracks.
Right. The tau gets phosphorylated. We call it PINO. It chemically changes shape. Those railroad tracks just collapse. And the tau tangles up into these toxic neurofibrillary tangles inside the neuron.
Which naturally leads to the final stage, the N, neurodegeneration.
Exactly. The neurons with collapsed tracks die, the brain tissue shrinks, and you start seeing cognitive decline, memory loss, confusion. So A leads to T, which leads to N. It was always viewed as a straight line.
But our source material throws a massive wrench into that straight line. I mean, the researchers looked at those resilient centenarians, and they also looked at a specific subgroup of 80-year-olds who had the brain pathology but no dementia. They labeled that group OCT-DEM.
And in both of these resilient groups, they found a massive discrepancy. They had a substantial amyloid burden. I mean, their brains were absolutely full of these sticky A plaques, yet zero dementia.
Which is wild, because the strict ATN model dictates that all that amyloid should have inevitably sparked severe tau tangles and neurodegeneration.
Right, but looking closer at the octogenarian cohort, the researchers realized that amyloid load wasn't the primary correlate of dementia at all. The true culprit was the phosphorylated tau, the POW. The 80-year-olds who were actually living with dementia had significantly higher break scores.
Okay, wait, let's pause there for a second. Break score is a term we hear a lot in neurology, and I want to make sure everyone listening really grasps it. What exactly is a break score?
Sure. So a break score is a staging system from stage 1 to 6 used by pathologists to measure the geographical spread of those toxic tau tangles throughout the brain.
So it's like tracking a fire spreading across a map.
That's exactly it. In early break stages, like stages 1 and 2, the tau is confined just to the memory centers, like the entorhinal cortex. But by the late break stages, 5 and 6, the tau has spread like a wildfire across the entire neocortex, which is where it starts affecting language, reasoning, and personality.
And the 80-year-olds with dementia had those high break scores.
Yeah, meaning rampant tau spread. And they also had incredibly dense neuritic plaques.
Right, neuritic plaques being amyloid plaques that are heavily intertwined with dead and dying nerve fibers filled with that toxic tau. So let me try to map this out with an analogy for you listening. Am I on the right track if I think of the old ATN model like a forest fire? Amyloid is the match and tau is the dry brush.
I like that, yes.
So the assumption was always, you strike the match, the forest inevitably burns down. But what this paper shows is that in these resilient people, there are matches dropping all over the forest floor. But the fire just doesn't start.
That is the perfect setup. The matches are everywhere, but the catastrophic tau fire isn't catching.
So before we jump into how they solved this, let's just do a quick recap of where we are. Number one, Alzheimer's isn't always a straight line from amyloid to tau to brain death. Number two, resilient brains can be full of amyloid matches, but they somehow avoid the tau forest fire. And number three, tau spread measured by break scores is the real driver of dementia in 80-year-olds.
Spot on summary. And to figure out why the fire doesn't start, the researchers had to confront a huge limitation in how we've traditionally studied brain tissue.
You mean the blender problem?
Yes, the blender problem. Historically, we focused heavily on identifying what cells and genes are involved in Alzheimer's, but we've been entirely blind to where those cells are physically located in relation to the pathology.
Which brings us to the methodology. And honestly, the technology they used here sounds like science fiction. They use something called Visium spatial transcriptomics and xenium in situ hybridization.
It really does sound sci-fi.
Right. So if you are not a molecular biologist, the old way of doing this, single-cell sequencing, is basically like putting a brain sample into a blender. You get a biological smoothie. You can analyze the smoothie and say, okay, there are blueberries and bananas in here. You know all the cellular ingredients.
But you've completely lost the original structure.
Exactly. You have no idea if the blueberry was sitting right next to the banana or on the opposite side of the tissue.
And that spatial context is literally everything in biology. So, to build on your analogy, what Visium and Xenium allow us to do is look at the brain not as a smoothie, but as an intact pizza. We aren't blending anything.
We're looking at the whole pie.
Right. With Visium spatial transcriptomics, we use these tiny microscopic barcodes. We can read the genetic activity of cells while keeping them in their native physical coordinates. We can look at the pizza and see exactly where the pepperoni, the amyloid plaques, is sitting relative to the cheese, the brain cells.
And Xenium.
Xenium takes it even further by offering single-molecule resolution. We're talking about mapping the exact geographic coordinates of millions of individual cells and the specific disease pathology immediately surrounding them.
Okay, so we finally have a microscopic map that lets us look at the exact forest floor. What did this map actually reveal?
Well, by keeping the pizza intact, the researchers focused heavily on a major battleground for Alzheimer's: cortical layers 3 through 5 in the superior frontal cortex.
That area is critical for executive function and decision-making, right?
Absolutely. And by analyzing the spatial neighborhoods of these cells, they identified 6 distinct tissue domains, which they labeled TD0 through TD5.
Now, these aren't just random clusters, right? They represent a physical spatial timeline of the disease progressing across the tissue.
Precisely. TD0 and TD1 are your pristine forest, homeostatic, healthy brain tissue. But as you move into TD2 and TD3, you enter the early adaptive response zones.
So what's happening in TD3?
In TD3, the environment is intensely rich in amyloid plaques. The matches have been dropped. And here we start seeing early glial inflammation. The brain's immune support cells, the microglia, are waking up and reacting to the plaques.
But then we cross the border into TD4. And from what I understand, this boundary between TD3 and TD4 is the literal physical inflection point we've been searching for.
What's fascinating here is that it is the moment the microenvironment in the brain fundamentally shifts. TD3 is driven entirely by amyloid. but cross over into TD4, and the environment is certainly dominated by POA.
The tau tangles.
Yes, it shows severe cellular damage and neuronal death. The tissue literally transitions from adapting to the amyloid to being actively destroyed by the tau.
It's the physical border where the match finally catches the dry brush on fire. For you listening, just imagine what this means on a human level. The microscopic difference between a TD3 environment and a TD4 environment is in the macroscopic difference between an 80-year-old who might, occasionally misplace their keys and an 80-year-old who can no longer recognize their grandchild's face.
It's a devastating threshold to cross.
It really is. So let's do another quick recap before we plunge into the mechanisms. 1. Visium and xenium let us map the brain like a pizza, not a smoothie. 2, the researchers found 6 tissue domains showing the disease timeline. 3, the boundary between TD3, the amyloid zone, and TD4, the toxic tau zone, is the critical inflection point of dementia.
Perfect.
Yeah.
And that leads us to the ultimate question. What is happening at this border to cause this massive shift?
Right. Why does the fire start?
The researchers found that the key lies in the brain's frontline immune responders, the microglia. They discovered this massive network of genes tracking this microglial response, which they called plaque-induced genes, or PIGs for short.
PIGs. That's a great acronym.
It is. Specifically, they isolated a gene co-expression module named OCT green-yellow. Think of this module as the master control board for how these immune cells react to the plaques.
Okay, here's where it gets really interesting. Because if we are looking at this control board, we see that the immune response isn't just one monolithic reaction, is it?
Not at all. It's actually broken down into 2 incredibly distinct phases, the early pigs and the late pigs.
Let's dive into phase 1, the early pigs. What are these microglia actually doing?
The early pigs correspond to a specific state the researchers labeled MIC5. This state is driven purely by the presence of amyloid. It is an adaptive cleanup response. When microglia sense amyloid plaques, they turn on genes related to the complement system and the tremin-2 pathway.
I want to make sure we explain those terms fully because they are so crucial. The complement system is basically the immune system's tagging mechanism, right? Let's use a mnemonic: paintballs, ingestion, garbage disposal.
Oh, I love that. PIG. Paintballs, ingestion, garbage. That's a great way to visualize it. Yeah. Think of the complement system, specifically genes like C1QB, as a crew running around with paintball guns.
Shooting the plaques.
Exactly. They shoot biological paint onto the misfolded amyloid proteins. This tags the plaques, it opsonizes them, to use the technical term, so the microglia know exactly what to attack and eat.
And what about TREM2?
Meanwhile, TREM2 is a receptor on the surface of the microglia. It acts like a radar dish scanning for this tagged amyloid debris. Once it locks on, it triggers the cell to ingest the garbage.
So genes like C1QB and TROBP are revving up the garbage trucks. They are actively trying to clear the tissue. And this early pig state peaks right in that TD3 environment we talked about. They are working hard cleaning up the amyloid matches.
Right, but then we cross the inflection point. We cross into phase 2, the late pigs.
And things get ugly.
They do. The late pigs map to a totally different microglial state called MIC2. Now, this state emerges when both amyloid and p-tau are present in the tissue. This is no longer a cleanup crew.
What are they doing instead?
This is a highly inflammatory antigen-presenting state. It's characterized by a massive upregulation of HLA genes and genes involved in lipid metabolism.
Let's break those down, too, for the audience. Why lipid metabolism, and what are HLA genes doing in this specific context?
Well, lipids are fats, and cell membranes are made largely of lipids. When those toxic tau tangles cause neurons to die and literally rupture, they leave behind a massive amount of fatty debris.
So the microglia have to eat all that fat.
Exactly. The microglia have to radically alter their internal metabolism just to digest all those dead cell membranes.
And the HLA genes?
HLA stands for human leukocyte antigen. When a cell expresses high levels of HLA, it is taking pieces of the destroyed tissue and displaying them on its surface. It's like waving a massive warning flag to the rest of the immune system, shouting, "Look at this damage. Attack anything that looks like this."
So it puts the entire area on high alert.
Yes, and this late pig state absolutely dominates the severely damaged PD4 environment.
Okay, so I have to ask a question on behalf of anyone listening who is tracking this immune shift. When these microglia transition into this highly inflammatory, high alert late pig state, are they the bad guys?
That's a huge debate in the field.
Right. Like, are the microglia trying to help but accidentally making things worse by going into overdrive and causing friendly fire? Or are they just completely overwhelmed by the sheer toxicity of the tau tangles?
That brings us to the crux of the cellular phase hypothesis of Alzheimer's. The data suggests that the late pig state isn't inherently evil. The microglia are simply doing what the immune system is programmed to do when it encounters severe intractable damage.
But they're causing collateral damage.
Yes. By transitioning from innate cleanup, just quietly taking out the trash, to chronic antigen presentation, they absolutely escalate the situation. This creates a state of sustained, localized inflammation.
And that inflammation is what actually kills the brain cells.
Exactly. The constant release of inflammatory chemicals ends up destroying surrounding healthy synapses. In the octogenarians, when this late pig state locks in alongside tau accumulation, it correlates incredibly strongly with severe neurodegeneration and dementia.
Wow. Okay, time for recap number 3. 1, the immune response has 2 phases. Early pigs for cleanup and late pigs for high alert inflammation. 2, early pigs use TREM2 and complement paintballs to clear amyloid. 3, late pigs are triggered by tau, they wave HLA warning flags, and they cause friendly fire that destroys synapses.
Perfectly summarized.
Which leads us to a massive lingering question, and really the ultimate twist in this story.
Here we go.
If this late pig inflammatory state is so tightly linked to tau spread and cognitive destruction in 80-year-olds, what on earth is going on in the brains of the resilient 100-year-olds, the centenarians doing the Sunday crosswords?
Let's bring the focus back to that centenarian profile, the 20 individuals in the CEN cohort. We know they have massive amounts of amyloid plaques in their brains, but we also know they have very minimal p-tau.
The matches are there, but the fire hasn't started.
Right.
So logically, you would assume their immune system just stays in the phase 1 early pig cleanup mode, right? Like, if there's no tau fire, there's no reason to trigger the late pig alarm.
That is the logical assumption, absolutely. And it's what the researchers likely expected to find. But they found the exact opposite.
Wait, really?
Yes. And this is arguably the most shocking revelation of the entire paper. The centenarians do activate the late pig programs. They strongly express those exact same antigen-presenting, highly inflammatory genes like HLA-DRA.
Wait, hold on. I need to make sure I'm hearing this right. They have the highly inflammatory late pig response, but no dementia. How does that not destroy their synapses like it does in the 80-year-old?
We connect this to the bigger picture.
Yeah.
It comes down to a vital biological concept called uncoupling. Yes. In the centenarians, the late pig response is triggered by the amyloid alone. They activate these genes extremely early in the disease process. Because of this, their immune response is completely uncoupled from the downstream accumulation of tau.
They have physically detached their microglial immune response from the toxic cascade.
Exactly. They uncoupled the train. The engine, the immune response is still running, but it's no longer pulling the train cars full of toxic tau explosives.
That is unbelievable. And the study highlights one specific gene that perfectly illustrates how this mechanism works, right? The SPP1 gene.
Yes, SPP1, which codes for a protein called osteopontin.
When I was reading the source material, this part completely blew my mind because it proves that in biology, context is literally everything. The same gene can be a hero or a villain depending entirely on when it shows up. Can you explain how osteopontin behaves differently in these two groups?
Gladly. So in the 80-year-olds who develop dementia, SPP1 turns on late in the disease timeline. It activates along the tau axis right in that highly damaged TD4 environment.
So it's showing up to a raging fire.
Right. And in that context, osteopontin acts as an inflammatory beacon. It binds to integrins on the cell surface and recruits more angry immune cells to the area. This promotes chronic inflammation and physically destroys the synapses connecting the neurons. It is highly detrimental.
But in the centenarians?
In the centenarians, SPP1 turns on early. It activates along the amyloid axis long before tau ever has a chance to accumulate. And in that early context, the mechanism is entirely different.
How so?
Instead of destroying synapses, the early osteopontin appears to help encapsulate the amyloid plaques. It literally forms a protective barrier around them, safely walling them off from the healthy tissue.
Same exact gene, completely opposite outcome based purely on the timing of its deployment.
Exactly.
Let me try to contextualize this with another analogy for the listeners. Think of SPP1 and this late pig response like a high-tech security alarm system in a house.
Okay.
In the 80-year-olds with dementia, the alarm system is wired incorrectly. The alarm only goes off when the house is already fully engulfed in a tau fire. It's too late. The alarm blaring is just adding deafening noise to the chaos while the structural damage is already happening.
Right, the friendly fire.
But in the centenarians, the security system is calibrated flawlessly. The alarm goes off the exact second a trespasser, the amyloid, steps foot on the front lawn. The response happens so early and so effectively that the trespasser is dealt with, the plaques are walled off, and the fire never has a chance to start.
That captures the mechanism perfectly. The centenarians aren't magic, they don't possess a muted immune system, and they certainly aren't immune to developing the initial amyloid plaques.
They just have amazing timing.
Exactly. Their profound cognitive resilience comes entirely from the incredibly precise timing and trajectory of their immune response.
Okay, final recap for everyone keeping track. Number 1, centenarians survive because their immune response is uncoupled from tau. Number 2, they activate the late pig alarm early in response to amyloid alone. And number 3, genes like SKT1 wall off plaques if activated early, but destroy synapses if activated late.
A perfect summary of a very complex mechanism.
So we've walked through spatial transcriptomics, tissue domains, microglial shifts, and uncoupling. Let's bring this all home. For the listener who is thinking, okay, this microscopic mapping is fascinating, but how does this help us actually beat Alzheimer's? So what does this all mean?
It fundamentally changes our entire approach to therapeutics. For decades, drug development was essentially just trying to sweep up amyloid or tau after the fact.
Which hasn't worked out so great.
No, it hasn't. But now we have a physical, mapped target, that A-beta tau inflection point between TD3 and TD4. This tells us that timing is absolutely everything.
Which means we have to administer treatments before that inflection point is crossed.
Exactly. If we're developing drugs that modulate microglial transitions, like anti-amyloid immunotherapies, or drugs designed to boost that TREM2 garbage collection pathway we discussed, we have to give them early.
What happens if we give them late?
If you wait until the brain is already dominated by late pigs and severe tau in that TD4 environment, the therapy won't just be ineffective. Pushing the immune system further when it's already in a state of chronic toxic inflammation could theoretically worsen the damage.
We have to fix the alarm system before the fire starts.
Precisely.
It's a massive paradigm shift from just get rid of the plaques to actively spatially managing the brain's immune timeline. The late pig state isn't a disease in itself. It only becomes harmful under specific pathological configurations. Knowing when and where to intervene is the true key to preserving human cognition.
The spatial map gives us the coordinates. Now we just need to deliver the right intervention at the exact right time.
Which leaves me with the thought that I literally cannot get out of my head since reading this research. And I want you, the listener, to really ponder this.
What is it?
We've just learned that the difference between losing your memory at 80 and doing the crossword at 100 isn't about avoiding the disease. It's about how your immune cells time their response to it. So if our brain's immune timing is truly the key to Alzheimer's resilience, could we one day vaccinate or train our microglia?
Oh, wow. That is a profound question.
Right. Like, could we develop a prophylactic therapy that teaches our immune cells to deploy that protective SPP1 response early, essentially reprogramming our brains to act like the brains of these centenarians, to automatically ring the alarm while the trespasser is still on the lawn?
Based on these findings, reprogramming that microglial timeline definitely represents the next great frontier in neuroimmunology. It's theoretically possible.
It's just wild to think about. We started this deep dive looking at a scan of a 100-year-old brain, a brain loaded with disease, full of plaques and pathology, but belonging to someone who is still living a vibrant, sharp, fully engaged life.
A true paradox.
Exactly. For decades, that scan was a paradox that broke all the rules of medicine. But today, thanks to spatial mapping, we know it's not a paradox at all. It's a blueprint for resilience.